My name is Ahmed, Senior Research Fellow at IIUM. Firstly, I would like to thank the American Council for Medicinally Active Plants International Conference Committee for this invitation. Our topic today is about use gum arabic methanolic crude extract and its anti-cytotoxic effect on human breast cancer. So the question is, why is this study so important? We know that the breast cancer cell line is the leading diagnostic cancer in women, and we found that the American Cancer Society stated about more than 200,000 new cases per year. This is almost about 30% increase annually, whereas more than 41,000 are the USA population estimated deaths annually. Also, the World Health Organization estimated that about 84 million people die from cancer each 10 years. Thus, it is constitutes that a severe public health problems in both developing and developed countries. So the current statement protocol include radiation therapy, surgical intervention, and chemotherapy, which induce a lot of side effects such as nausea, fatigue, vomiting, weakness of innate immune system, as well as hair losses. So some studies have confirmed that polyphenols isolated from Acacia Seyal gum or from gum arabic have a several potent biological activity such as anti-inflammatory agents, controlling the cardiovascular disease and renal failure, as a recent study of renal failure protocol 2021. So the major bioactive compounds thought to be the responsible for this activity or biological compounds are phenolic acid, flavonoid, terpenes, lignins, and alkaloids. Despite the anti-proliferative effect of gum arabic was studied on several biological properties, however, it did not consider the study of breast cancer behavior using based on a chip technology platform. Therefore, this study focused on cytotoxic effect of methanol crude extract as the optimum crude extract. That means having a lot of bioactive compounds thought to be very beneficial for killing breast cancer. The objective of this study to study proliferative effect against breast cancer, MCF7 cell line exactly, properties, and extracted from the natural and commercial sample, namely Prebio-T commercial sample drawn from the same kinds of Acacia Seyal gum using the in vitro methodology. So to achieve these objectives, we have a novel method here in these graphs called the graphical step demonstrates the follow of SRP as a novel protocol for anti-proliferative detection, the source adapted by the OSA. So in the main result, we found that as well, we see here the methanol MCF7 breast cancer cell line validity at 100%. We found that hand-picked select sample called Acacia Seyal gum crude extract, we found that the IC50, that means inhibition concentration that kill 50% of cell lines, about 9.5 microgram per ml. This amount from the crude extract, the cells and the solution phase can kill 50% of the cell line with log GI. This gross inhibition almost close to 1, and the R square also close to 1. If you compare the hand-picked select sample, we found to the commercial, we found that it is at least there is a significantly difference Here is a little bit smaller. We found that IC50 8.7 is a little bit difference. The lowest IC50, that means the most potent or the most active. Also, it is clearly, can see that here, clearly can see that here, if we compare the normal cell, see the control density of the normal cell with the dead cell. Here is very clear between two of both them. This demonstrates the image show the potent surviving MCF7 cell line at 24 hours incubation, showing the treatment of acacia gum for both sample crude extract from the methanol crude extract respectively. In this area here, this figure, this is a proposed mechanism of action of how Acacia Seyal gum inhibit the MCF7. This is what we do right now. You can see this clearly, because no time we can't speak a lot regarding this topic. So in the conclusion, the result provides scientific evidence of the beneficial effect of Acacia Seyal gum, thus adding a body of knowledge to the gum Arabic research. This work also will serve as a paradigm or new platform for the value added Acacia Seyal gum or gum Arabic in general industry beyond the food ingredient domain that will bring socio-economic benefits of the nation. And finally, we find that the methanol crude extract of gum Arabic considered being a novel defense-based agent for the prevention and cure of breast cancer. For the current and future work, we need to use this. This is currently we do this one. We found that the isovitamin C is a main bioactive compounds resistant on the gum Arabic. So we need to do this on the Brest chip technology. So finally, this research is supported by we acknowledge really for this research supported by protocol for postdoctoral research merit program from the Islamic Development Bank. And the author also acknowledged the International Institute of Halal Training Biotechnology Engineering Department, UIA facilities. And finally, special thanks also for Prof. Nuruddin Ashmakha, University of California, Los Angeles, USA, for unlimited and academic support and orientation to the main author. Also, our knowledge extends to the Perfect Life company for offering the commercial sample. Thank you very much once again for to be able to be with you on this conference today. And really, I'm very happy if you have the question to clear everything's not mentioned here in this short time. Thank you very much.